Hello people, my name is Judith Constantine from Dar es Salaam, Tanzania. Welcome to my YouTube channel. I facilitate pharmacognosy-related subjects at MUHAS, and this is part 1 of plant tissues of diagnostic importance. We will cover the introduction to plant tissues of diagnostic importance, tissues and structures of diagnostic importance, other specialized structures and inclusions, the importance and diagnostic application of plant tissues. So please, let's get along. Now, in pharmacognosy, the microscopic analysis of plant materials relies on characteristic plant tissues and specialized structures for the identification, quality control, and authentication of crude drugs. So these tissues are very, very important when it comes to authentication of plant sources. So the plant tissues of diagnostic importance are tissues that are analyzed in the laboratory to determine a variety of plant health issues and for structural identification. If the plant has been through stress or something, there are some tissues that can help us identify this kind of stress. If the plant is from a certain source or a certain family, there are some tissues that can help us identify all those. So we will go together through this and see how we can navigate about it. So plant tissues are specialized cell groups performing specific functions. Apart from being used for diagnosis, they have functions in plants. They include the dermal tissue system, which is specialized for protection. They include vascular tissues. These are meant for transport. Sometimes they are grouped into vascular bundles. There are ground tissues which are meant for metabolic support, and each of these will be discussed separately for us to follow and understand. So understanding plant tissue organization reveals why plants respond to pruning, to environmental stress and propagation techniques, and many other issues that the plants are likely to go through. Now we will start with the dermal tissue system, which is composed of the epidermis, and within the epidermis we find trichomes and stomata. Each of these will be discussed in separate sections. So in this section we are going to see the epidermis in general, then we will come to specific tissues as we move forward. Now, the dermal tissue system is the plant's outer protective layer, which is analogous to the human skin, primarily composed of the epidermis. So the dermal tissue system is the very outer part of the plant. And the main part of the dermal tissue system is the epidermis. And the epidermis covers young plants and regulates water and gaseous exchange through stomata and trichomes. These two are going to be discussed in separate sections, separate parts, maybe part 2, part 3, and many other parts as we go. So in all the woody stems and roots, the epidermis is replaced by the bark, which is a tougher protective layer, the epidermal cork, that also controls gas exchange through lenticels. The main function of the dermal tissue system is protection, water regulation, and absorption of nutrients and gas. So essentially, like I said earlier, the epidermis is the main component of the epidermal tissue system. And sometimes the epidermal tissue system is felt as the epidermis. The epidermis consists of a single layer of cells covering the whole plant. It is the waxy protective layer whose thickness and surface features can be diagnostic. We are going to see how the thickness, how the surface features can help in diagnosis of various plants. The epidermis is mainly composed of two, the trichomes and the stomata. But all these are within the epidermal cells. we are going to see about epidermal cells and how they are specialized to perform specific functions. The periderm is a secondary protective tissue replacing the epidermis in woody plants. So when it comes to woody plants now, we don't call it the epidermis anymore. It is the periderm because it has been replaced by something thick. So typical, like I said something about specialization. So typical epidermal cells are often devoid of chloroplasts. They are not specialized to perform any function apart from that of the epidermal cells. This is different from specialized epidermal cells. For example, the stomata guard cells. Those are epidermal cells that have been specialized to perform in opening and closing of the stomata. There are subsidiary cells. Subsidiary cells are also specialized. They are epidermal cells, but they have been specialized to perform specific functions. They help the guard cells when opening and closing the stomata. We are going to see as we move because subsidiary cells will be discussed along with stomata because they play a big role in diagnosing different types of stomata. We shall see this, and I don't want to talk about it in this part. Epidermal cells show great variety in form, giving characteristic patterns when seen in surface view. In transection, they are often flattened, parallel to the surface, and square or rectangular in shape. I will show you in the image. The outer walls are often convex and most markedly thickened. The epidermis of the stem and the trees and shrubs is usually destroyed early by development of the cambium. I've said this earlier, but on the stems of herbaceous plants and leaves, fruits, and seeds, the epidermis persists and often yields highly diagnostic characteristics. We are going to see these. The structures of the epidermis and stomata are of first importance in the microscopic identification of leaves. When it comes to leaves, we deal mostly with the stomata and the epidermal structures. This is the microscopic view of our onion cells. We said it is kind of rectangular, sometimes concave. So this is how normal and specialized epidermal cells appear. Once they are specialized, they will appear differently from what we are going to see as we move. This imaging, please, I recommend you to purchase the book of Trees and Evans, the 16th edition. It has all this. These are epidermal cells in various medicinal plants. But I would like you guys to see how epidermal cells, we cannot discuss epidermal cells without discussing the trichomes and stomata, because they appear together. The trichomes and epidermal and stomata are within the epidermal cells. So these are surrounding epidermal cells in various types of stomata. We shall see the types of stomata as we move. They may look like something weird here, but they have a meaning. For example, we have anomocytic stomata, we have actinocytic stomata. We shall see all this as we move. Don't stress yourself. But these are the epidermal cells, and they can be specialized. As you see, the more the specialization, the different the appearance. So these are normal and specialized epidermal cells surrounding stomata, but these ones have already been specialized because they appear differently. The subsidiary cells have been specialized. So we have straight-walled epidermal cells. These are seen in Jaborandi, Foca, and Senna leaves. Please go and read more about this. We have wavy-walled epidermal cells, which are seen in Stramonium, Hyoscyamus, and Belladonna. We have beaded walls that are seen in lobelia inflata and digitalis lanata. We have papillus, epidermis, which is seen in leaf of coca. So please go and read around this. So these are the images. These are straight walled, wavy walled, straight wavy, slightly wavy walled. We have beaded, and we have the papillus, like that which is seen in coca. So there is a thick cuticle which is developed in aloe leaf and the bearberry leaf. I said earlier, this is characteristic of most of xerophytic plants, the plants that can persist in drought. We have striated cuticle, which is found in belladonna, jaborandi, digitalis lutea, and digitalis tapsi. Sometimes the epidermal cells the epidermis may have inclusions like mucilages, like calcium carbonate oxalates, calcium carbonate crystals, spherocrystals of diosmin. These are inclusions. Remember, we said something about inclusions in the objectives, and here is where we get to discuss this. Mucilage is present in the epidermis of senna and butu leaves. Cystoles of calcium carbonate occur in epidermal cells of Urticaceae and Cannabinaceae. Spherocytes of diosmin occur in butu and epidermis. If you can watch on diagnostic substances, we discussed much about the calcium carbonate crystals and the families that are mostly have calcium carbonate crystals. We have about 5 families, so please go and read about this for more understanding. You see here, for example, here in A, we have the tissue, the epidermal cells. See, these epidermal cells should occur in this structure, but once they have the cystoletes, the epidermal cells appear more larger, like outgrowths of cells. They appear larger than what we should be expecting in normal epidermal cells. And here we have epidermal cells of Rhizoma betulina, which shows spherocrystalline masses of diosmin within epidermal cells. So the epidermal cells have inclusions. And here we have epidermal cells that have inclusions. These include, remember we did something about hyaluron grains, something about crystalloids, about globoids. So please go and watch part 1 and part 2 of diagnostic substances that has been posted a few days ago. And here we have a lot, so please go through your book of Trees and Evans, try to see along. Like here we have inclusions in epidermis. These are starch grains. Remember we did something about starch grains. Yeah. And here I said you guys should read about inulin. This is inulin deposited in the epidermal cells of some plants. So that's it. And you see a sunken stomata here. We will see what a sunken stomata means as we go through stomata. So don't stress yourself. Just go read about what you have done today on the epidermis.